Okay, so we are on to lecture 15, which is our digestive system third lecture. And we're going to continue on. We just finished the stomach. So we are about to move into the small intestines, the large intestines, the rectum, and the anus. So starting off, the small intestine is actually the largest section of the GI tract. Even though it's called small intestines, it's definitely not small in its length, but rather in its diameter. The small intestines collectively is about 20 feet long in the average adult, but it's only about an inch in diameter if you were to measure that. Transit time, by far, this is where the majority of our ingested substances reside, because this is where we're actually going to finally break down into the tiny absorbable units what we've ingested. So this takes a lot of time to meticulously, chemically target all of those bonds between our ingested foods so that we can result in monomers. So those are the individual absorbable units of monosaccharides and amino acids, fatty acids, and glycerol. So we'll do the majority of absorption at this point, and essentially the completion of digestion. So transit time through the small intestines may take 5 to 6 hours, as opposed to the stomach where we may spend maybe about 2 to 4 hours. All right, so I have here site of digestion, completion, and absorption. Most of the absorption takes place at this point. We do have absorption taking place in the stomach, but the majority of that is going to be some of the stuff that we ingest that is absorbable already. So lots of medicines actually get absorbed in the stomach. We also have some vitamins get absorbed in the stomach. But usually proteins have not had enough time to get broken down. Even though the stomach is the first place where protein digestion begins, it only begins there. And we still have a lot of breakdown that needs to continue in the small intestines. So by the time you get to the small intestines, you're going to have a lot of nutrients that have finally gotten broken down to the point where they can actually get absorbed. When we look at the small intestines, one of the things that's most notable is if you were to move the serous membrane that's kind of covering all of the vasculature, this is a very vascular tissue. In order to absorb all those nutrients, you not only have to take them across the mucous membrane of the GI tract, but also, of course, into blood vessels so that you can distribute them throughout the body. So in this drawing here, you can see just how amazingly vascular the small intestines is. Just, the red and the blue here represent the arteries and the veins. So you have arteries coming in to the tissue, and then veins that are carrying out all of those amazingly absorbed nutrients that have just passed through that small intestine. So it's very, very rich in blood supply for absorbing and then transporting those absorbed nutrients. The first place most of them will go, in fact, is going to be the liver. In fact, all of it will go to the liver. All right, before we actually get into the organs of the small intestines and large intestines, I want to talk a little bit about the supportive structures. So remember, the abdominal cavity is a very, very large space. The ventral body cavity is the largest space, and of that, the abdominal pelvic cavity is the largest space, single space in our body. We have a lot of movable viscera there. So the small intestines is moving, the large intestines is moving, the stomach is moving. Of course, even when we're breathing, although the liver itself doesn't move, when the diaphragm is moving, the liver is going up and down within the abdominal pelvic cavity. So all of our viscera not only has to be able to move relative to each other, so it has to slip and slide, it has to move smoothly against the inside of the ventral body cavity, but it also has to be secured because all of this viscera is actually quite heavy, especially when it's filled with our ingested substances and secretions, and you don't want it to end up in the bottom of your pelvis. So you actually have to tether all of that tissue down. And when you think about it, the only place that you can really tether or tie or support all of your viscera is to the stable supportive rod that runs the entire length of our trunk, and that's your spine. So if you look here in red, this is the serous membrane. So this is that nice slippery serous membrane that lines the inside of the body cavity. We learned this that very first lab. So this is that parietal peritoneum. As that parietal peritoneum comes around, we're going to see it's going to go all the way around the dorsal body wall. And when we get to the vertebrae, this is where you're going to see something really amazingly interesting occur, and that is that the serous membrane is actually going to come off the body wall posteriorly. It's going to go out to our viscera. And this, of course, is simplified. They're showing you one loop of small intestine, but it's going to come off the body wall, and that's going to wrap around the movable viscera and come back. And when it does that, what it's going to do is it's going to create this double serous membrane. This double serous membrane that we have here, I'm going to change the color so I can see that a little bit better. That double serous membrane we see here, this now, when you have two sides to it, we actually call that a mesentery. So it's two sheets of serous membrane that come together. That's going to do several things. The first thing it's going to do is it's going to help to stabilize that loop of intestines. So it's tied essentially to the dorsal body wall, so it doesn't fall to the bottom of your pelvis. But then it also will create this amazing passage for blood vessels and nerves to travel to and from our organs. So blood vessels, you're never going to see them traversing across the body cavity. You're not going to see a blood vessel traveling to an organ. They always move through what we call mesenteries. So these are these double sheets of serous membranes that come off the back of the body wall and go out to our organs and come back again. You're also never going to see tethering coming off the front of the body wall and going to an organ. They're always tied to the back. And that's because this is the only place where we have a consistent stabilizing structure that runs the length of the body cavity. So we're going to name a number of these mesenteries, but these are going to be basically just simply double sheets of serous membranes that come off the dorsal body wall. They're going to function to stabilize and support. It's still allowing some movement, and then also be a protected conduit or pathway for blood vessels and nerves to and from our organs and tissues. All right, so this is an artistic rendering of a mid-sagittal section of the abdominal pelvic cavity, and you can see here's the liver. We're going to tie the liver up against the inside of the diaphragm. This is actually referred to as a ligament, but it's really just that serous membrane, and it has a lot of connective tissue in it. And so anytime you have a serous membrane where the connective tissue of that membrane is filled with dense fibrous connective tissue, they often will call it a ligament. So that's what we're going to see here. We're going to see a number of those throughout the digestive system. But anyway, so this is what's called a falciform ligament. It kind of holds the liver on the undersurface of the diaphragm. We can also see here the serous membrane comes around, it goes over the surface of the liver, and it's going to come out here. That's going to go around the stomach. So this portion here, again, that's going to be a mesentery. You can see here coming off the dorsal body wall and going out to this piece of the large intestine. Again, this is a mesentery, so double sheet of serous membrane. Here you can see all these amazing loops of the small intestine. Remember, there's like 20 feet of it. So again, that serous membrane is going to come off the dorsal body wall, and it's going to go around all these organs. And where you have this double sheet, we call this a mesentery. And again, we're going to have blood vessels that get to travel through those, and nervous tissue that's going to travel through those mesenteries. So it's these wonderful pathways, or we call conduits, protected conduits, pathways for blood vessels and nerves. So they also are going to be protected inside that pathway, inside that mesentery. So they also aren't going to get twisted or broken away from the tissues that they're supposed to be supplying. All right, so this is a really nice photo looking at a loop of small intestine. This one's beautiful because what I can see is this kind of yellowy sheet here. It's shiny. This is the serous membrane that covers on one side. You'd have a serous membrane on the other, and then kind of sandwiching in between those two serous membranes is adipose. That's what has this yellow coloring. So you kind of remember Serous membranes are like saran wrap. So think of it this way. You have a piece of saran wrap that's going over the surface here and then back around on the other side. And where the two layers of saran wrap come together here, we're going to put between those two layers adipose tissue, and then we're going to run our blood vessels through there as well. The adipose tissue just helps support this network of blood vessels so they're somewhat more stabilized in that mesentery. So this drawing over here is basically drawing what we see here in this photo of a loop of small intestines. Obviously, the surgeons are holding that loop out. Here, what they did is they cut off one side of that serous membrane so we can see the blood vessels that are running in between that double serous membrane or that mesentery. So I have here, our functions are to stabilize the relative positions, right? They help to prevent that entanglement so they don't tie up on each other. It's pretty amazing because you can surgically go into someone's abdomen, lift out all these small intestines and just kind of put them back in. When I was in grad school, I did surgeries on rats where we were moving intestines around. And I remember my major professor just said, well, just push them back in. And I was like, well, don't they have to go in a certain way? And she said, no, because the mesenteries will keep them already in their relative position so they don't actually get disoriented or entangled. And then last, conduit. A conduit just means a pathway for blood vessels and nerves. Okay, so you can see here, it provides a pathway to and from the organ that's protected. So again, you're not going to see blood vessels traveling into any region of the small intestines from the body wall other than through these mesenteries. Okay, so again, providing protected pathways to our tissues. All right, so again, they're secured to the dorsal body wall. So if you notice, all of these pieces of the GI tract and accessory structures are all secured essentially to this dorsal body wall. Right? We don't see anything coming off the anterior body wall. So this is nice and smooth. If you open up the abdomen, you basically can cut through the musculature and skin of the abdomen, and you just open up this big space, and all the organs, tissues sit inside, and everything is going to be attached to that dorsal body wall. So in this photo or drawing here, what they're doing is they're pulling the small intestines anteriorly. You can see that it's kind of stretching here. This is the mesentery because it is attached to that dorsal body wall. Same thing with this loop of the large intestines. This also has a nice mesentery. You can see the blood vessels in it. And again, if you tried to pull it out, it would get tight as it's also attached to the dorsal body wall. All right, histologically, the small intestines has a unique feature that are called plicae circularis. I mentioned them briefly in our very first digestive lecture. So these are macroscopic folds, meaning you can see them with the naked eye. So in this photo here, this is a endoscopic picture of the inside of someone's small intestine. So there's no magnification really that they're using to see these. These basically circular folds that go around the length of the tube rather than down the length. These circular folds are what they call pleats. You can think of them as pleats within this mucous membrane. I like to think of them as running down the tube. I'd be doing little hurdles over these, so you're kind of jumping, jumping, jumping. Remember, rugae are more like slides. They go down the length. But plicae are going to be at 90-degree angles to the long axis rather than parallel to the long axis. So these are macroscopic folds of just the mucosa. If you look at the outside of the tube, it's smooth. The inside of the tube has these folds. So these are folds just of the inside mucous membrane. All right, and they're pulled into those folds again by that muscularis mucosa. Okay, so this is looking at some of that histology again. Notice here, this is the muscularis externa. So notice the outside of the smooth intestine. Small intestine is going to be smooth. So it's going to be a smooth tube. But what we're going to do is take the mucous membrane and we're going to fold it up into these pleats. Okay. So what this is trying to show you, this right here, this is the muscularis mucosa. There's this little line of muscle there. Okay. That's the muscularis mucosa. And so everything above this is the mucous membrane. Everything below this is the submucosa. So in this histological image, what you're looking at is this big fold here. If I can draw, stay on my That's the muscularis mucosa. I'm trying to stay on it, right? And so this nice big finger-like fold here, macroscopic, this is a plica circularis. This is this big pleat or pleat in the mucous membrane. Okay, so this is what we call a plica circularis, this big upward fold. It's kind of like if you have a rug and you kick the rug and it creates this big fold in it. But maybe that rug has some little tiny piles in it or whatever. You know, like you can see here that has little tiny texturing on the surface. We'll talk about that in just a second. But these big folds or pleats are unique to that small intestine. So the idea is, remember, anytime you see folding, and this was true for our motor unit, remember we looked at the motor end plate, where we had the sarcolemma at the motor end plate folded, right? So we had a lot of surface area for our neurotransmitter receptors. Here we're going to fold the membrane so that we have, again, a huge surface area, not for receptors, but for absorption. We're going to have to use protein channels to absorb the majority of what we have eaten. And those protein channels have to be in the membrane. They have to be in the cell membrane. And so we have to have a lot of membrane surface. And so we're going to start folding the intestinal mucosa. So this is the first layer of folding that we see in the intestines. All right, so we're going to use the mucosa and the submucosa to kind of fold up into this macroscopic feature, okay? All right, then what we're going to do is we're going to continue to fold. So in this drawing, this is a plica circularis, this nice big fold. So again, it's kind of like the rug kicked up into a big pleat or a fold. So these are plica circularis, but you can see that this mucosa is textured. It's kind of, again, like having a rug that also has a little pile to it or texturing to it. These little tiny finger-like folds are just of the mucosa itself, so not including the submucosa. These are just the mucosa itself, and these are called villi. So villi are finger-like folds of only the mucosa, where plica circularis are going to be the mucosa and submucosa. You can see here how the submucosa gets pulled in to the pleat, right? So this is the submucosa getting pulled into the plica circularis. A villi is just in the mucosa. So we have these teeny tiny little finger-like folds of just the mucosa. So that's the epithelium and lamina propria. So rugae and plica circularis are macroscopic folding, and then the villi are going to be a microscopic folding. And we can kind of see texturing. So if we could look at a cadaver closely enough, when you look at the cadaver's small intestine, or living small intestine, you can see it looks kind of fuzzy. You can't quite see clearly the villi, but you know it's not smooth. It looks a little bit fuzzy. This is a histological image. And again, what they're showing you, this big fold here is a plica circularis. And then there's another one over here. This is a big plica circularis. And then these little guys coming off are villi. They don't look quite as uniform as we see over here, and that's because they're soft. And so they're going to be at slightly different angles. So when they make a slide, again, you have to kind of remember these are soft, so they're kind of like noodles and trying to slice noodles. They aren't going to cut really evenly. So these are the villi, though. So these are these little finger-like folds of just the mucosa. So that's the lamina propria and the epithelium. So these would be the villi. All right, so again, the idea of the villi is to further increase folding. So in this really nice simplified drawing, we have a plica circularis again, which is pulled into this pleat by the muscularis mucosa. And we're going to pull the mucosa and submucosa into this pleat. And then we're going to see that the plica circularis is still further folded by folding the mucosa itself and these little tiny villi. So here's a macroscopic view of just the mucosa. At this point here, you're looking at the muscularis mucosa, and these little finger-like folds, these are the villi, right? So you have these little finger-like folds of the epithelium and the lamina propria. So the idea is to increase, again, surface area, and they estimate that you have about almost 3,000 square feet of mucous membrane when you're looking at that overall folding that occurs. All right, so this is a photo inside the small intestines, and you can see here these are little tiny villi. So again, it looks kind of like a shag rug here. You have like these little tiny finger-like projections. So these are, again, what we call villi, and you can kind of see the plica circularis here, these nice big folds. So you have these sort of circular folds going at a 90-degree angle to the tube, and then you can see the texturing at the surface. Okay, so this is a quick video on villi structure.
In our body, absorption of food mainly takes place in the small intestine. The inner wall of the small intestine is highly folded and has many small projections called villi. The structure of the villi is specially adapted for food absorption. The villi are finger-like and large in number. These features greatly increase the surface area for absorption. Moreover, the epithelium of the villi is only 1 cell thick. This provides a very short distance for diffusion of food molecules into the blood. The cell membrane of the epithelial cells has a large number of microvilli. This further increases the surface area for absorption. Each villus has a lymph vessel called lacteal in the center. The lacteal is surrounded by a network of capillaries. These allow the absorbed food molecules to be carried away rapidly. Therefore, a steep concentration gradient of food molecules can be kept across the wall of the small intestine to increase the rate of diffusion of food molecules.
Okay, so when we look at one of those little villi, like in the video they just showed us, there's 2 really important structures that sit inside each villi. Remember, the idea is to increase the surface area for absorption. And obviously, we're absorbing not just across the epithelium and into the connective tissue, but of course, what resides in connective tissue is blood vessels. And those blood vessels will be able to then distribute as they absorb and then carry away the nutrients and water that we've just consumed and absorbed. So we're going to see a tremendous amount of vascularity within each of those little tiny villi. And then we're also going to see a structure called a lacteal. A lacteal is a fancy name for this ended lymph vessel. So when we look at the lacteal, it's going to have its starting point here at the beginning portion of the villus. So it's going to start here, and it's going to absorb and pull what it's absorbing into this lymphatic system. So when we talk about absorption, what's really important to recognize is that size of your absorbed molecules makes a big difference. Things that are tiny, monomers, can absorb into blood vessels. And so I don't know if you remember from our very first digestive lecture, the amino acids, which are going to be the breakdown of our proteins, those are going to be small, and these can get absorbed across the epithelium and then also into our blood vessels. So these can get taken away. So we're going to absorb amino acids. Also monosaccharides. So if we have monosaccharides, I don't know if I'm not going to spell all monosaccharides, but let's put monosaccharides, monos. These are also small. So these are your glucose and fructose and galactose. Those get absorbed because they're also small. So things that are small, so these would be amino acids from our proteins and our monosaccharides from our carbohydrates. those can get absorbed very easily across the epithelium and into our blood vessels. But when we look at our fats and our oils, those are not water soluble. And so the fats have to be resynthesized into a structure called a chylomicron. And it's going to be basically a structure that is combined with some protein by these epithelial cells. So the fatty acids are going to get absorbed into the epithelial cells. But from here, we're going to make them into these chylomicrons. They're actually going to be quite large. And those are too big to fit into our blood vessels. And so we're going to need to absorb these chylomicrons into our lacteals. So our fats and oils actually get absorbed into our lacteals because lacteals as lymph vessels are very permeable. They have larger gaps and spaces. And so that's how we're going to absorb our fats and oils versus our proteins and our carbohydrates. Those get absorbed into blood. Fats and oils get absorbed into lymph. So I'll show you a slide of that in just a second. All right, so again, we look at blood vessels. These absorb our sugars, right, our monosaccharides, and our amino acids, which are a breakdown of protein. And our lymph vessels, specifically called lacteals, when they sit inside these little villi, these are the closed-ended lymphatic vessels. So unlike blood vessels where we have an entry point and then we have an exit point, so this is a continuum, the lacteals start in these little villi, and then they'll pull the lipids, which are going to be our fats and our oils. Oops, I can't really do this, but fats and oils in from our ingested substances that have gotten broken down. All right, so here's a much prettier slide of that. So here you can see a carbohydrate getting broken down. It's going to break at these glycosidic bonds eventually until we have monosaccharides. So again, these monosaccharides are small enough. We're going to have little protein channels here, glucose channels or GLUT channels. These glucose channels are going to be able to absorb the glucose, and these are small enough that they're then going to move through another protein channel here and then move into our blood vessels. These little blood vessels actually have gaps between their endothelial cells, so they'll be able to absorb these small monosaccharides. So we have carbohydrate absorption. Same thing happens for our proteins. These are made up of amino acids. We're going to break the little peptide bonds here till eventually we get to amino acids. These also will have little protein channels here. We'll also see protein channels here. And once they get through the cell membrane, they go into the blood vessel again through these small little gaps in the endothelial cells. So these get absorbed. So protein into blood, carbohydrate into blood. But over here, remember, these are not water soluble. So that's the problem. So they're not water soluble, which means all of the digestive substances that we use, all these enzymes that we use, is basically secreting a large amount of water. So it's basically mixing oil and water, which don't mix very well. We're going to emulsify, so add a little soap essentially. So we break them down into small lipid droplets, but they're still fatty acids, which are going to be easily about 20 carbons long. These are still pretty big. They'll be able to diffuse across the cell membrane, so we don't need a protein channel here because they're lipid soluble, but we can't carry them in the fluid of our body. So we're going to make this substance called a chylomicron, and so that's going to be basically fatty acids plus protein, and that'll be made by the epithelial cells. So that will be able to then make this water soluble. It basically has polar proteins that sit on the outside. Now these now are too big to go into our blood vessels, but lymph vessels have bigger gaps and spaces in them. And so that can absorb this really big chylomicron. And so the lymph vessels are used to absorb the digestive products of fats and oils. Okay, so this is a quick video on that.
The lining of the small intestine is characterized by numerous circular folds called plicae circularis. The plicae are lined with finger-like villi. From a cross-sectional view, the villus contains a network of capillaries which surround a specialized lymphatic vessel known as a lacteal. The epithelium of an intestinal villus consists of columnar cells which are covered with microvilli. This succession of folds and projections increases the surface of the intestinal lining for efficient absorption. Carbohydrate digestion is completed by enzymes in the small intestine. Carbohydrates are absorbed by the villi and then enter the capillary. Fat digestion occurs primarily in the small intestine. Fat molecules are digested and absorbed into the epithelial cells of the villus. The fats are formed into clusters called chylomicrons, which pass into the lacteal. Lymph carries chylomicrons away from the villus. Protein digestion is completed in the small intestine. Proteins are broken down first into peptides, then into amino acids. These are absorbed into the villi, then into the capillary.
Okay, so that was a really nice animation. You can play that again, but it shows you how we absorb our carbohydrates, our proteins, and our fats. So you really need two absorption structures. We need a network of blood vessels or capillaries, and we need the ended lacteals. So those are going to start in the villus and then travel to circulating blood. So eventually all that lipid we absorb does get deposited in our circulatory system. We'll talk about that a little bit later, but that's going to actually happen in the cervical region. So we're going to have a union between our lymphatic vessels and our blood vessels. Okay, so here we can see, this is really great. This is showing us blood coming from one of the villus. So inside of here, of course, are going to be carbohydrates and our amino acids. And we're going to see that where all of the blood goes from the GI tract, and this is all of the blood, which is crazy, we're going to see go straight to your liver. So all of the absorbed digested monosaccharides and amino acids that get absorbed by our intestines is going to go straight. And this is actually not just by the small intestine, it's also the large intestines and the stomach. All of this absorbed nutrient is going to travel directly to the liver before it goes anywhere else. So you have to move through the liver before you can go anywhere else. So from the liver, we're then going to go into the heart, and then the heart can start to circulate those nutrients out. So the liver is going to be an amazing nutrient sort of regulatory center. So it's going to take raw nutrients and that's going to metabolize them in the liver before we send out sort of regulated nutrient blood. When we look at the lymphatic vessels, so these are these big chylomicrons, they don't go directly to the liver. They're going to bypass the liver and they're going to be dumped into the circulatory system up here in the cervical area where you can see an entry point into blood. So at this point, they travel into blood and then go to the heart. They'll travel around the heart and eventually they'll end up in the liver. So you have this very roundabout way for ingested fats and oils to eventually get metabolized by the liver. The liver's job is going to be nutrient regulation. So its job is going to be to regulate all of our nutrient load. And it does not just for carbohydrates and proteins, but also our fats. We just don't end up in the liver directly. We have a very roundabout way simply because it's not water soluble. The chylomicrons are going to be too big to go directly to the liver. Okay, so this is looking at a side-by-side, a drawing, and then a histological image. So this is looking at a small villus. So you see a little villi. Notice here's the columnar cells. And remember, these are going to be very, very active in absorption and secretion. So you have nice, simple columnar epithelial cells. Underneath these, this is the lamina propria. And the lamina propria is going to house the blood vessels and lacteals. Histologically, it's very difficult to see those. They are single cell-walled structures. But in here would be a lacteal traveling right up the center. Can't really draw it very well, but there's a lacteal. And then you'd also have blood vessels going around the lacteal, and all of this would be absorbing the nutrients that have been broken down in your lumen of your um, small intestines into either the lacteal or the blood vessels. And then, of course, these nutrients, if it's in the blood, would be on its way to the liver. And if it's fat, it would be on its way to circulating blood and then a roundabout way back to the liver. Okay, and so this is what drawing here. You can see this is right in the center, that's a lacteal. So this is a closed-ended tube. So it's going to start here in the villus, and it's going to be absorbing our ingested and digested fats and oils. And then you have the blood vessels sitting around. They're going to absorb the amino acids and the monosaccharides. Okay. So one last layer of folding that we're going to see are the microvilli. So microvilli are further folding that occurs. And here we're looking at one of the epithelial cells of the mucous membrane. And you can see that the cell membrane itself is folded. And this is what we call microvilli. Microvilli are the microscopic foldings of the epithelial cell membranes that make up that mucous membrane. And again, the idea is further folding to increase the surface area for absorption and secretion. Okay, so this is looking at the layers of folding that we see. So this is that progressive increase in folding. So this right here is showing you a plica circularis. And again, these are macroscopic foldings, including the mucosa and submucosa. Then we have the little villi. This is the folding of just the mucosal membrane. And then we have, and that's what this is a villi. And then we have the microvilli, which are going to be these little tiny microscopic foldings of the mucous membrane's epithelial cell membrane. Okay, all right. So when we look at the small intestines, it has 3 regions to it. From the outside, it's very difficult to tell where one region starts and another one stops. But histologically, there's some very important differences between the 3 regions that we're going to look at. But the 3 regions, the first portion is called the duodenum. That's about a 10-inch distance from here to here. It's what we call the C of the duodenum. So you can see how it makes this letter C. It's a very short segment. It's the shortest segment of the small intestine. And its job is really all about functioning to help neutralize the acidic chyme. So here's the stomach. The stomach, remember, has secreted hydrochloric acid from the parietal cells. So the pH is about 1 to 2, so it's extremely acidic and corrosive, so it can burn easily through cell membranes. So the first thing you want to do is help to neutralize that. So as this acid chyme comes in, first of all, the duodenum has to be prepared for that, so it's going to have a lot of those mucus cells lining it. But we're going to find that the pancreas, which is right here, so this is the pancreas, this is going to secrete sodium bicarb that helps to neutralize. And then we're also going to see, can't quite tell here, but this is the common bile duct. And this is also going to have bile, which is very neutral. And that's going to help to neutralize this acid chyme. We're going to find that the acidity in the duodenum can go from about 1 to 2 as it enters to about 8. So the pH can go up to actually a little bit basic by the time you're done moving through this very short segment. And that allows the rest of the small intestines then to complete digestion and absorption of what we've eaten. Okay, so then we have this next segment here, which is about 8 feet of small intestines, and that's what we're going to refer to as the jejunum. And then the remainder is the ileum, and the ileum is going to be about 12 feet, and that's the last segment before we get into the large intestines. So we have the duodenum, which is about 10 inches, the jejunum, which is about 8 feet, and then we have the ileum, which is about 12 feet. Okay, so the duodenum is the shortest segment. Again, it's about 10 inches. Duodenum supposedly means 12 fingers. So I think that's how they measured it originally. It was about 12 fingers long. It forms the letter C. So you can see here, this is the duodenum right here. They've cut it open so we can see inside these nice little plique circulaires here. Sitting in its medial border, we're going to see that it actually holds a really important glandular tissue. This is the pancreas that is going to be intimately related to the duodenum. The pancreas is going to be primarily exocrine. It's going to secrete a lot of pancreatic enzymes and chemicals, one of which is sodium bicarbonate. You can see these little tiny openings here. There's one here, and there's one here. These are called ampullae. And this is where the pancreatic duct is going to be able to release its secretions into the small intestine. So that's going to happen here and here. And we can see this green tube behind, that's the common bile duct. And it's going to merge with the pancreatic duct to secrete bile into the small intestines here at the duodenum. So the duodenum is going to be receiving basically acid chyme from the stomach, plus bile from the liver, and pancreatic juice from the pancreas. So it's really going to be this kind of mixing bowl of chemicals occurring within this very small segment of the small intestines. All right, so at the entry point of the small intestine, so where we see the end of the stomach and the beginning of the duodenum, this is the pylorus right here. That's that really thick muscular area. It's going to regulate the movement of this acid chyme into the small intestine. Here we can see a cross-section of that pyloric sphincter, so really, really thick muscle. They estimate you get about 1 to 2 teaspoons of acid chyme moving in at a time into the small intestine. So it's a very slow movement from the stomach into the small intestines, because we want to make sure we have a small enough amount of acid chyme to really start to break down the small bonds between the monomers. This is a endoscopic image of the inside of the small intestines looking up at that pyloric sphincter. All right, so here we're looking at some of the functions, and this is a really nice artistic rendering, but it's really a mixing bowl of chemicals, like I mentioned. You're going to be receiving acidic chyme. So remember, chyme is just the term that's describing what we used to call the bolus. So that's your ingested substances with salivary amylase in it, right, saliva. Once you move into the stomach and you're adding all that acid, then we change the name from bolus to chyme. So chyme is really what you've ingested at this point and have partially digested. So we're going to receive that. That's going to go in from the stomach. And then from the small intestines, pancreas, and liver, we're going to get enzymes and chemicals. So we can see here the liver and its accessory structure, the gallbladder. This is bile. Bile is pigmented green, so that's what we're looking at here. So bile is going to be coming into the small intestine. We have our stomach acids and chyme coming into the small intestines. And then we have coming from the pancreas, enzymes and chemicals. So we have basically stomach, liver, and gallbladder combined, and then pancreas all dumping into this very first segment of the small intestines of the duodenum. All right, so histologically, the duodenum is very, very distinct. Again, this is one of my favorite drawings, but this pie piece here is showing you what the small intestine would look like if you're looking at just the duodenum. And again, remember, the GI tract is basically 4 layers or 4 tunics, and every area of the small or the GI tract has those 4 layers. But the reason we call one layer, for example, the duodenum and one the jejunum, is how those 4 layers are modified. So here we're going to say we do have plicae, so it's going to increase the surface area. It's not really a large plicae, it's kind of a small plicae. But in the submucosa, we have all of these what we call duodeneal glands, or we call them submucosal glands because they're sitting in the submucosa, or they're also called Brunner's glands. These are making mucus, and they're just basically secreting it onto the surface of the duodenum's mucosal layer, and that's going to prevent erosion coming in from that acid chyme, until we have enough time to neutralize it, we have to protect that mucous membrane. And so this very first segment, because it's exposed to so much acid, has to protect itself by secreting lots and lots of mucus. Right, so that's one of the major features. It's really easily identifiable by those duodenal or submucosal glands. All right, so here's submucosal or Brunner's glands or duodenal glands. Basically what we're looking at is, here's the mucosa. So we're going to see it has villi. In this view, I can't see oblique, but we have villi out here. And this is the lamina propria in between. This is all submucosa, and you can see these sort of circular structures. These are all these nice mucus glands. So mucus glands, remember, even on a low magnification, it's relatively easy to tell because you can see their columnar cells. They have nuclei around the outside edge, and it's kind of a clear, glassy, cytoplasmic filled cell. So that's all the mucus that they're secreting. All right, and then again, the function of all this mucus, basically production and secretion, is to protect the mucous membrane, the inside of the duodenum, from the acid coming from the stomach. All right, and then this is just another histological image. And again, we see villi, so these are villi out here. And then this is the muscularis mucosa right there. And so all of this down in here, these are submucosal glands. So these are all these nice Brunner's glands. So this is all secreting lots and lots and lots of mucus. And then here we see this is the muscularis externa. All right, the jejunum's our next segment. By this point, we would have finished all of the neutralization of the chyme. The chyme at this point has now been neutralized to a pH of about 8, so it's even a little bit basic by this point. So we don't have to protect anymore against the acidity. So this is really all about digestion and absorption. So the major feature here is just tons of folding, really, really big, long plica circularis, really, really, really long villi. So when I look over here at this inset image, notice that this plica circularis is really big, and we have really long villi. And that's going to allow us to have a huge surface area, lots of area for blood vessels to enter and get some of that absorbed nutrient to circulate, as well as those lacteals. So this is primarily where we're going to finish digesting, and we're going to see a majority of absorption taking place here. It's about 8 feet long. Obviously that varies a little bit, but about 8 feet in length. So quite a long distance for absorption to take place within. All right, histologically, again, really, really, really large. This is what we're going to look for: large plica circularis and really, really long villi. So that's what we see here. This is a huge plica circularis. Look at how long this guy is. Super long plica circularis, right? So this is the submucosa, and there's the mucosa, and really, really long villi. So nice long villi. So we have a lot of surface area for all of that absorption to take place in. All right, and then this is just another image of that. This is the muscularis externa. This is submucosa. So notice I don't have a lot of glands in here. And they just have these really, really, really long villi. Okay. All right, and then the ileum is the last segment. Ileum supposedly means twisted, and this is about 12 feet long. It's the last segment. It tends to be a little bit more twisted or packed in the inferior aspect of the abdominal pelvic cavity. This is where we're really going to finish off nutrient absorption. By the time you're done with the ileum, essentially your GI tract is done digesting and absorbing what our bodies have the biochemistry to do. So we've absorbed everything that we've broken down at this point, and then we're going to enter into the large intestines. So this is the last section of the small intestines here. And you can see how it comes in to this medial edge of the large intestine. So this is the beginning of the large intestine. And the job of the large intestine is going to be to house an amazing microbial flora. And the microbes that reside in our large intestines are essential. We need them. Their job is going to facilitate the breakdown of nutrients that we don't have the biochemistry for, releasing some really important vitamins and essential fatty acids. And at that point, once the bacteria have broken down some of those nutrients and vitamins, we can then absorb those. So that occurs in the large intestines. So by this point, with the ileum, we have finished the absorption of anything that we have the biochemistry to do. And we're going to then move into the large intestines, where we're going to allow our microbes to take a very important role in releasing nutrients and vitamins that we otherwise wouldn't have access to. All right, so the entry point into the large intestines from the small intestines is called the ileocecal valve. And the reason for that is because this portion of the large intestine that is inferior to where the small intestine comes in. So this is where the small intestines comes in. So this is the opening right here from the ileum into the large intestines. This segment below is called the cecum. So you can see this is the ileum coming into the large intestines on this sort of medial border to it. The cecum is this enlarged pouch that sits below that with the little appendix coming off the bottom. So the function of this ileocecal valve or sphincter is to prevent the reflux and migration of bacteria into the small intestine. So we don't want that. I want to make sure that bacteria don't go this way, right? So we're going to prevent that by having this ileocecal valve. If bacteria get into the small intestines, the problem is that there's a lot of nutrients still that we are trying to absorb into our own bodies that we don't want our microbes to have access to. So we need to protect the small intestines from that refluxing of bacterial load. Okay, so if we look at the ileum, we're going to see that the ileum has in its small intestine submucosa a very important structure. Now, we have lymphoid tissue throughout the small intestine, but in the ileum, which is that last segment, we're going to see huge aggregations of lymphoid structure. So similar to our tonsils, we're going to see large areas of white blood cells. The difference in the tonsil is that the tonsil, the lymphoid structures are going to be surrounded by a layer of connective tissue, so they're going to be dissectable as distinct individual structures. Here, they're just sitting in the connective tissue, so they're not really going to be surrounded by it. So we're just going to see sort of mounds of lymphoid tissue, mounds of lymphocytes or white blood cells. And so these are going to help fight off any of our own bacteria that are trying to migrate backwards into our small intestine. So as opposed to our tonsils, which are lymphoid structures protecting us from bacteria coming in from what we're eating, the lymphoid structures here in the small intestines are preventing a sort of our own bacteria from getting into places they shouldn't be. So these are called Pear's passage or lymphoid nodules rather than tonsils, where again, a nodule is just really an aggregation of white blood cells sitting in connective tissue, but not bound and surrounded by connective tissue. All right, so these lymphoid nodules are we call Pear's patches, right? Here you can see where it says PP, that means Pear's patches. What you're looking at is all this dark purple. That's because lymphocytes are primarily just a really large round nucleus with very little cytoplasm. So when there's a lot of them, it ends up just looking like this big mound of purple because all the nuclei stains purple. So you have all of these individual mounds of white blood cells, and these are called lymphoid nodules or Pear's patches, and they sit in that submucosa. So this is the muscularis mucosa right here. This is the muscularis externa right here. And so they're sitting in this submucosa. And you can actually have so many of these Pear's patches that when you look at the surface of the small intestine, and I'll show you a picture of this in a second, it actually looks a little lumpy from the outside. Most of the small intestine just looks like a smooth tube, but where you have a lot of Peyer's patches, it looks a little lumpy at the surface. Okay? And again, the significance is that we're protecting ourselves not from bacteria coming in from what we're eating, but from resident, beneficial bacteria that we don't want to migrate into areas of our small intestine. Okay, so we're preventing these resident bacteria from entering into the small intestines from our large intestine. All right, and so this is a histological image, and here you can see just this really big purple mound of white blood cells sitting in this submucosa. We still have, again, villi up in here. So we still see villi. We're still going to see a little bit of plica circularis, but the identifying feature are Peyer's patches. Anytime you see these big purple nodules in a large aggregation, that's going to indicate we're in that last segment of the small intestine. All right, and then I want to talk a little bit about those mesenteries that we began with. And if you watch the cadaver video I posted, I'll show you images of these. But this is looking at the abdomen if you were first to open it. So this is basically cutting open down the linea alba and then removing the oblique muscles and the rectus muscle and the transverse muscle of the abdomen. And this is actually what the viscera looks like when you just simply take off the abdominal wall. There's nothing attached to that abdominal wall, so it easily kind of comes off. You can see that here. And what we're looking at is this nice big sort of adipose-laden curtain that kind of lays over the small intestines here. This curtain is called the greater omentum, and it's attached to the greater curvature of the stomach, so this outside edge of the stomach, bottom and outside edge. And then also the transverse colon. You can't quite see it here, but the large intestine is going to be right here. In this image over here, they've basically taken this really nice long sort of adipose-laden curtain and they've kind of lifted it so you can see it's actually attaching. This is the large intestine here to the transverse colon, the large intestine, and the stomach. This adipose-laden sort of draping here is called the greater omentum. And it's going to sit right on top of or anterior to the intestine. Again, it's attached to the greater curvature of the stomach and the transverse colon. And it helps to maintain the position of the small intestines, but more importantly, it helps to insulate. All of this viscera is very, very active and generates a lot of heat, and we don't want to lose all that heat through our anteroabdominal wall. Typically, your abdomen shouldn't have a lot of fat in that subcutaneous tissue right under the skin, and so we want to insulate that with visceral fat. So this is all visceral fat here in the greater omentum, and that helps to insulate the abdominal viscera. All right, in addition to the greater omentum helping to support and reduce sort of the movement of the small intestines, we also have, more importantly, the mesentery proper. So the mesentery proper is going to be this mesentery that's coming off the dorsal body wall, going out to the small intestines and then coming back. It's going to help support and limit the movement and entanglement of the small intestines, as well as being that conduit for blood vessels and nerves. And again, here's an image of that mesentery proper with all these blood vessels traveling between that double serous membrane. And again, the adipose that sits in between those kind of sheets of saran wrap or serous membrane is going to help to support these blood vessels. Okay, so the large intestine is the last segment of the gastrointestinal tube, right? So this is that tube that goes from mouth to anus. And this is the last segment. It's about 5 feet in length in the average adult. And it consists of several different regions. So we're going to start off where we have the cecum down here below. We have the colon, which is going to be the ascending, transverse, descending. Then we have what's called a sigmoid colon. And then we'll have the rectum and anus. Okay, so this is the last segment of our GI tract. The colon is actually really cool when you look at it because it creates a framework. It's like a board or a frame that goes around the small intestines. It also helps to confine it. So going all around the small intestine is going to be the colon. All right, the colon's job is really important. Probably the most important thing is going to be to absorb the tremendous amount of water that exists within the small intestines. They estimate we have about 10 liters of water moving through our small intestines in a day. 2 liters of that is going to be likely ingested as part of what we eat and drink, fluid from the foods and the fluids that we ingest. And about 8 liters is actually going to be what gets secreted through the course of a day. So we need to make sure that we reabsorb all that fluid. Diarrhea, which is, that really loose stool, results when you have irritation or infection in the small intestines and colon, where you're not absorbing the water that you would have secreted. The problem with not absorbing that water is that if we don't maintain our blood volume, because that water is coming from your blood. If you don't put that water back into the blood, your blood pressure drops, your blood volume drops, and we're no longer able to circulate fluids, right? And so those fluids then don't go to the brain and other important organs. So one of the primary causes of death in third world countries is actually diarrhea, because you lose too much fluid. So we need to make sure that we absorb that water. So that's actually going to happen here in the colon. So it's this water reabsorption. In that water, of course, are electrolytes. So those are going to be the minerals like sodium and potassium, those things that are charged molecules that are dissolved in that water. We also are going to absorb some of the vitamins and nutrients that were liberated by our microbes that weren't available in the small intestines that are now available because of the microbes breakdown. And then compaction. We need to make sure that we squeeze down what's left over that we couldn't absorb into appropriate, easily removed fecal matter. Again, those microbes are going to be really important in finishing off any digestion that could have happened by the small intestines but didn't, because you don't have the biochemistry. And the microbes also can help coordinate immune responses. So this is just a fun little drawing. You can see here, these are supposedly the bacteria, and they're sitting there, doing their job of releasing some vitamins. So you have your vitamin B released by bacteria. They're going to help break down anything that didn't get absorbed at this point because it couldn't be broken down, and kind of cleaning off the gut. Okay. All right, this I just thought I'd throw in here. If you're taking microbiome at the time, this is kind of a neat drawing, but this is the major bacteria that are present in our GI tract. We do have some bacteria located within the stomach, so we have what we call Helicobacteria. These are able to withstand the acid chyme. We have some in the small intestines, so Enterococcus and Lactobacillus. But the majority, you can see here, sit within the large intestine. So this is the large intestine bacterial flora. You can see just a huge number of bacteria live within this large intestine. Okay, so a little bit on bacterial flora.
Have you ever wondered what it means to be human? Well, it turns out that only a tiny percentage of what you and I are made of is in fact human. And we need these non-human bits to survive. But we're doing dreadful things to this hidden majority, and it's damaging our health in ways that we are only just discovering.
It is said that we are 10 parts microbe and 1 part human. And that's counting microbial cells as the unit. If we count unique genes, then the difference is a factor of 100. We are only 1% human in terms of unique genes.
So you and I are mostly bacteria of some kind. We need each other to stay healthy, and there's now a name for this part of ourselves.
The human microbiome is a set of microbial communities that have co-evolved with us and we with them, and have become part of the human landscape. It is an extension of self. They provide to us untold functions that we don't have the means of executing. We're born deficient in our ability to digest certain kinds of foodstuffs. For example, the complex polysaccharides of plants. We can't manage those very well. Our microbes provide us the means of taking advantage. They also help us manage lots of other encounters with the environment.
One of the things our microbiome does is give us an internal shield of friendly microbes which protect us against invasion by some of the nasties out there. The good guys try to crowd out the bad ones.
It's competitive exclusion. Another term for it is colonization resistance. So, they man the beachhead and make it hard for others to arrive and find space.
So where are these microbes? Well, while most are in our bowel, they can in fact be anywhere where we're exposed to the outside world.
So we have them in our mouth. In the mouth, it's a measly 10 million per gram, but it's only measly because in the colon we're talking about 100,000 times as many per gram.
So the microbes in our bowel, for example, help us to recirculate valuable nutrients and in fact assist us to keep our cholesterol down. So we need them and they need us. We do some terrible things to our microbiome, like assault it with antibiotics.
Yeah, unfortunately for us and for them, everything I'm going to tell you about the untoward effects of antibiotics has to be balanced against the obvious important benefits. But I think for too long we have felt as though you can't do much wrong with an antibiotic. So if in doubt, pull it out. And that clearly is not a good position to be taking. We've been asking volunteers to take an antibiotic even though they didn't really need it. We do it for short periods of time. We watch them before, we watch them after, and we're now doing second exposures a half year later. And the bottom line is, maybe not surprisingly, far more members of the microbial communities of the human body experience a severe impact. They're decimated, many more than we thought.
Now, in healthy people who might only have an occasional course of antibiotics, the microbiome seems to recover quite well. That's not so true in people who are sick and weakened. As a side effect of antibiotics, they can become infected with a germ called Clostridium difficile, which causes hundreds of thousands of deaths worldwide.
So you're right. So Clostridium difficile is the cause of large numbers of deaths, but even larger numbers of illnesses, where there is a recovery, but after some cost.
One solution that some people suggest for this devastation to our microbiome is probiotics, substances which put back the friendly bacteria.
The evidence on probiotics is still early to interpret, but I would say it's suggestive that there is some potential benefit there.
As I said before, these friendly bugs, our microbiome, does a lot more than protect us against nasty infections. They keep our cholesterol down and detoxify poisons. So the question is, if we damage our microbiome with antibiotic overuse, are we causing heart disease and cancer?
Short answer is we don't know, because no one has looked for this particular link in the story. We simply don't know.
So while in the past we've seen bacteria as the enemy and got excited about how we can kill off disease-causing germs with antibiotics, it's time for a new way of thinking. Remember, we're talking about 99% of what we are.
Okay, so anyway, I thought that was a really great clip showing us the importance of our microbiome. All right, so the last thing I want to do is look at the regions of the large intestine. So again, we have our cecum, which you can see here in this drawing, is this region that is at the beginning of the large intestine. So we're looking at this area just below where the ileum comes in. And then we have the colon, which is consisting of the ascending colon, the transverse colon, the descending colon, what we call the sigmoid colon, which is kind of this S-shaped portion, and then the rectum and anus. Okay. All right, so the cecum is what we call the blind pouch. Blind in the sense that it basically is not starting anywhere. I mean, it's not a continuation of anything. So you have it starting here. You have the appendix being this little diverticulum coming off of it, usually about 6 centimeters. And then you have this blind-ended pouch here. In some organisms that actually do a lot of fermentation, like rabbits, for example, they eat a lot of fiber. They use their bacteria to help break that down. And so it ends up being a very, very large structure. In us, because we don't do a whole lot of what we call hindgut fermentation, we don't need a very large appendix. So some people will say it's vestigial, we don't need it, but it's laden with bacteria and immune structures that were used for fermentation in other organisms quite heavily, but not quite as much in ourselves. So we have this pouch-like region inferior to the ileum entrance. The entrance into the cecum from the small intestines is this ileocecal valve that prevents that reflux. Okay. All right. So the appendix, about 6 inches, or about 3 inches, or about 6 centimeters long. It's a finger-like extension. It's filled with lots and lots of lymphoid tissue. And then you have here this appendix. You can see this little finger-like diverticulum. Inside would be a lot of lymphoid tissue within the mucosa. Again, a lot of people say that it's vestigial, meaning we don't need it because our bacteria, of course, are laden throughout our large intestine. So you don't really need the appendix. And sometimes what happens is if something gets blocked, like sometimes it could be a seed or something that you've eaten that hasn't gotten digested, that sometimes can block this, or you have a contortion of it. So depending on the position of the appendix, it can get blocked. If bacteria are inside of here and fermenting, they create a lot of gas as a byproduct. And that can actually cause the appendix to rupture if that gas doesn't have an exit point. So if there's torsion or something blocking this, and that can result in a ruptured appendix, where then you can get the bacteria migrating throughout the outside of the gut that can lead toward sepsis of the individual and death. So it's really important if you have an appendicitis where you have inflammation of the appendix to get that treated before you get a rupture. All right, so again, the appendix is really just an immune tissue here, has just lots and lots and lots of lymphocytes laden within it. Okay, 4 regions of the colon. Again, we're going to see we have the ascending colon, transverse colon, descending colon, and sigmoid colon as those 4 regions. And in this drawing, you can see those outlined here. Ascending is going to go up on your right side. We're going to have a 90-degree turn here. That's what's called the right colic flexure. We're going to see that the colon goes up on the left, and that's because the liver sits on the right side of the abdominal pelvic cavity. So there's more space on that left side. So it goes way up on the left side here. This is the left colic flexure, and then it goes all the way down as the descending colon. This is now in the curvature of the pelvis, so it runs on this inside curvature of the pelvis, and then it goes up right down the center of the pelvis here. So this is the sigmoid colon, and then goes down in through the pelvis as the rectum, and then, of course, the last exit point would be the anus. These flexures are also named according to the organs they're next to. So the right colic flexure is also called hepatic flexure, and the left colic flexure is also called the splenic flexure. All right, so again, I have ascending colon going up the right side, transverse colon going from right to left. So this is going to go essentially right underneath where the stomach is. Descending colon goes down on the left side, and then that sigmoid colon is really sitting in the curvature of the pelvis here, so it can get to the center of the pelvis. So sigmoid colon has that sort of S-shaped look to it. And then we have the rectum. The rectum is just collecting feces. So it's a collection space. And when it stretches, with a fairly large amount of feces here, that then signals a readiness for defecation. All right, features of the large intestines. One of the features is going to be the Taenia coli. Remember that the GI tract has the musculus externa, and you have that inner circular and outer longitudinal. This is really just the outer longitudinal layers. They get reduced into just little strips. We can see that here. So there's one of the strips on the outside. You can see another strip here and another strip here. The Taenia coli are basically just reductions in the outer longitudinal muscle, and they're shorter than the length of the large intestine, so it kind of pulls the large intestines into what we call haustra. So these are pleats or sort of gatherings of the large intestines. So these haustra are these compaction pouches. So they're helping to kind of squeeze out the water so that feces dries and compacts. So this is a nice cross section through the large intestine. And so in red, this would be the inner circular muscle layer. And in orange, this is the outer longitudinal. So we're going to reduce the outer longitudinal to 3 distinct strips. Okay. One of the important clinical applications to this is that because we don't have the outer longitudinal going all the way around, if you have someone that has a very slow moving colon, where you're going to be able to squeeze the feces down so it gets really hard, sometimes what can happen is this mucous membrane, so blue is supposed to be the mucous membrane, can sometimes get squished through the gaps between the circular layers. And since you don't have the outer longitudinal to protect that, you can actually get what we call diverticula, where you have a layer of the mucous membrane flipping backwards through a gap in the small intestines, the large intestines, inner circular layers, and that can cause diverticula and then can cause necrosis and infection. Okay, so this is looking at a large intestine. So they're showing you the cecum here. Again, this is the cecum. This is this blind pouch that ends in the appendix here. This is just a bunch of lymphoid nodules lading the inside of this diverticula of the appendix. We can also see really well the haustra. So these are these sort of compaction pouches. And I can see, at least in this case, 2 of the 3 layers of the Taenia coli. Taenia coli means the flatworm. A Taenia is a flatworm of the colon. Okay. All right, so again, the haustra are these compaction chambers or compaction pouches that help to squeeze down all the fluid out of the fecal matter and help remove water and make the feces a much more compact and easily defecated substance. This is looking inside with an endoscope at the large intestine's haustra. So these are these compaction pouches. This is looking at a radiopaque dye that gets put into the colon. So you can see here, this is the rectum. You can see how large and full the rectum is at this point. And the opacity is within the large intestine. So you can see here's the descending colon that goes way up on the left side. Of course, this is the ascending colon on the right side. Notice it's much lower because the liver is going to sit here. And this is the transverse colon going across. The stomach would be sitting right in this location. And you can see these compaction pouches. So these are the haustra. Okay. All right. Rectum and anus. Remember, rectus means straight. So we had the rectus abdominis and you had the rectus femoris. So rectus just means straight. The rectum is the straight portion of the GI tract that's going to exit directly down through the pelvis. So that's why it's referred to as rectum. It's straight. It's about 5 inches, and its job is to store feces until appropriate time of defecation. It has stretch receptors in it. So when the stretch receptors sense a significant volume of feces or appropriate volume of feces, that then signals a readiness for defecation, and we'll have an inner and external anal sphincter. When you get those stretch receptors going off, that's going to signal relaxation of the inner anal sphincter. And then you'll see the feces kind of drops a little bit lower. And then you have the option of either exercising and utilizing your external anal sphincter for defecation, or you can squeeze this off and you can prevent defecation until a more appropriate time. All right, so this is looking at the rectum and anus. Again, remember the rectum means straight, so we're looking at the straight segment here. Again, it's about 5 inches long, so we're looking at this last segment, and there's the anus. Again, here's the rectum, so you can see it can get quite full or quite large, and then the anus, so we have the inner and external anal sphincter there. All right, and this is the last slide, so this is a little video looking at traveling through the GI tract, and then we'll be done. Oh, done too soon. There you go. So this is actually an animation again. So this isn't the someone's real GI tract. They're animating what they think it looks like with an endoscope. So that's the small intestine as you're traveling through. Then you'll get to the lower esophageal sphincter. So this is the lower esophageal sphincter, and then we're going into the stomach. So there's supposed to be rugae. There's a peristaltic wave, right, in the stomach. That's supposed to be acid chyme there. We're going to go out the pylorus, so there's the pyloric sphincter. And going into the small intestine, so notice we go into the small intestine, we have the plicae circularis, so definite change in the mucous membrane there. So there's bile coming in, right? Bile and pancreatic juice. So those are coming in from the pancreatic and bile ducts. So this is all small intestine, so notice plicae circularis, so down the entire length. So we're in the ileum, or sorry, the jejunum. And then we'll move into the ileum. Round and round and round through the small intestine. Eventually getting to the ileocecal valve. All right, so this is the end of the ileum. There's our ileocecal valve, and now we're in the cecum. And now we're going up through the ascending colon. These are supposed to be haustra. We're in this nice big compaction pouches. Now going down toward that sigmoid colon, into rectum, and then the anus. Okay. Yeah, that was all sigmoid colon. So now we're in the rectum, and then that's supposedly feces, and we'd be getting ready to defecate at that point. So this would be the defecation.